In this video, we'll be talking about the trucker of all minerals, and that's calcium. Calcium, super important. If you have any problems with pest and disease, and who doesn't have problems with pest and disease, any quality problems, especially for the horticulture guys out there, if you have any post-harvest disease problems, super important you understand what's in this video. So really, pest and disease quality all comes down to calcium, and we're going over exactly what I mean by that today in this video. We'll be going over calcium in the plant, calcium in the soil, and how it all interacts. We're talking about application rates. When we talk about calcium, of course, you have to talk about liming and pH, so we're talking on that a bit. We'll be talking about fertilizer options, what to use and what not to use. We're talking about soil health, Cal-Mag ratio, balancing, and whether or not that's valid or not. And most importantly, we'll be talking about how to manage calcium for plants' health in a regenerative system. So if that sounds like you, watch the whole video, or you can find the chapters of the video. It's a pretty long video, so you can find the most important parts to your operation or you're interested in throughout the video. So that will be in the video thing below. So this is part of our regenerative nutrition management series. It's also in a course-like form on our website, so you can go check it out. It's completely free. There's not even an email opt-in, which is pretty cool. And effectively, I will be talking about everything I know about calcium from a regenerative point of view and even conventional point of view. And these are the type of things I implement on people's farms as a regenerative consultant. So let's get into it. Calcium. Calcium is referred to as the trucker of all minerals by a lot of people in the regen space. And it's a key driver of quality. So that's quality in your product as well as quality in growth. So it's super important. So in terms of its roles in the plant, there's three main roles. The first one is cell wall integrity and function. So calcium is important for developing the cell wall and membranes. The way this works is that in the membrane, there's these things called pectins. Now, pectins almost have a structure like this, a bit of a zigzag pattern. Now, if you don't have calcium, the plant's going to produce all these zigzag patterns, and they're all going to be mumbled up and with no structure to it. What calcium does is it can bind to two different pectins, causing them to come together in a bit more of a rigid form. So it brings them together like this. We've got our calcium in between like that. And because it has the two positive charges, it can hold one positive charge to one and then the other positive charge to the other, bringing them together. So in a real way, it's almost like when you have a cementing job and you put rebar in it, and that rebar adds strength to your concrete slab. In a very similar way, calcium acts as that rebar, and it reinforces and it brings everything into alignment, adds reinforcement to your cell wall. And that's super, super important because obviously your plants need cell walls, and that comes into two main functions. The first one is just the function of that cell wall. It's important because it creates a very rigid and strong structure for the plant, and that's important to prevent the losses of, say, water and solute from being lost from the plant. So if the plant's cell walls were very weak, you get this loss from the cell, so from within the cell into the environment. So evapotranspiration rate goes up because it's effectively losing this water through a weak wall. Or weak cell wall. With that function, it improves the translocation of minerals around the plant. So if you think of it like a road, you can't go very quickly on a road that's full of potholes. Likewise, if you have a nice smooth road, you can transport stuff really, really easily. So calcium does the same thing. It makes sure that all those transport systems within the plant are actually flowing properly. So a very important point that comes from this is just the functioning of the plant, but also quality. So if you think about a lot of the post-harvest problems, a great example of this is end blossom rot, which is effectively where there's not enough calcium in, say, a tomato. So we've got our tomato here, calcium deficient, and you get that black rot on the bottom. That's not so much actually caused by a disease, but it's more so that there's a deficiency in calcium which prevents a strong cell to be formed, or strong cell walls to be formed, allowing for microbes to then start to decompose the plant because it's not healthy. So this is very much a calcium deficiency and not really a disease problem. So quality draws quality, make sure that cell walls are strong enough to have a really nice skin. That's super important for post-harvest storage, making sure you don't get diseases. Also the firmness of the product, super important. We'll be talking about managing post-harvest diseases and other diseases later in the video when we talk about regenerative management. But right now, just keep in mind that cell wall integrity that drives this function. So that's quality. The other thing that's super important, and that's disease management, pest and disease management. So what happens is when you have a really strong cell wall in the plant, it prevents pests and diseases from breaking that cell wall to then enter into the plant. And so when we look at the Mineral Nutrition and Plant Disease book, there's a whole bunch of different diseases that calcium can increase the tolerance of the plant against. So these are a lot of things like mold, a lot of fungal diseases, which the increase in calcium prevents those fungal pathogens from breaking through the cell wall. Majority of the plant defense systems from calcium are driven by an increase in cell wall integrity. So this is super, super important. So that's a lot of like blights, rots, mildews, any really fungal pathogen. Calcium is a super important one to defend defend against those pathogens. So as I said, cell wall integrity and function. Calcium drives quality because it maintains a really good cell structure, keeping, say, your apples and your cherries and all that intact. They're not going to break. And the other thing is that it increases pest and disease resistance because they can't break through that layer, those cell walls. Next is cell division elongation. Now, calcium, for more or less the same reason as it increases cell wall structure, it's required in building those cell walls in new cells. And so calcium, the big driver of quality growth. And so if you think of nitrogen as you put nitrogen on and you get this really good crop response, that's good, but it's not quite quality. A lot of the times if you put nitrates on, it has very much a dilution effect and it just forces growth. Calcium, on the other hand, stimulates cell division and elongation, but it has the minerals to back it up for a strong cell wall, which means that when you do get a calcium response from these applications, it really stimulates cell division, but it's creating high quality cells. So when we look at this book, Mineral Nutrition of Plants, there's a really good reference in the calcium chapter about the relative growth of tomato plant and perennial ryegrass and calcium applications. So as effectively calcium concentration increases in the solution, perennial ryegrass and tomatoes grow more. And effectively you can double the growth rate with the correct calcium applications. Now, if you've seen the critical points of influencing yield video, you'll notice that cell division is very important for effectively all of the critical points of influence. So bud determination, as well as after pollination, you get cell division. So the embryo is dividing up and you want as many divisions as possible so you get the biggest fruit as possible. That's where cell division is super important. So we want to make sure that we've got a good calcium supply for really good cell division. The other really important thing is elongation. Now this is particularly important in roots. So in the book they explain a study that looked at the difference between root extension, so the length of the root, in a calcium deficient scenario and a calcium sufficient scenario. What they found is that in a calcium deficient scenario, root extension was about 5 millimeters long. In a calcium sufficient scenario, it was 20 millimeters long. So that's a 4x increase in root extension. Now, if you think back to the rhizophagy cycle, we want to make sure we get good root elongation to stimulate the rhizophagy cycle. That's super important. Now, just general increase in root biomass is really good because it allows the plant to explore more of that root zone, giving it more access to different minerals and nutrients and water. Super important. So if you can effectively get a 4 times increase in your root zone because you have sufficient amount of calcium, think of that's effectively 4xing the amount of nutrients available to the plant. Super important, super important. But in terms of rhizophagy cycle, and we'll be talking about this later in terms of the regenerative management, we want to have really good cell division because that's going to be happening on the tip of the cell, on the root. We want to make sure that our plants have that new developing root with elongation so that it can then take in the microbes for that rhizophagy cycle. If you don't know what I'm talking about, go check it out. It's a super fascinating concept in study, and it has some really big implications on how we manage nutrition. But it all comes back down to calcium. We need to make sure we have calcium to then stimulate that cycle so that we can then get all our other minerals through the rhizophagy cycle. Finally, there's this signaling effect. So calcium is known to be a signaling molecule in the plant. It doesn't really have too many implications except that it does help with signaling for pathogens, signaling within the plant when the plant is affected by pest and disease. Not too sure how this actually plays into management, but regardless, if we have enough calcium, then I'm sure our plants can then function properly. So all in all, calcium is super important for the whole plant to function, for quality growth. And effectively, I just got a little diagram here with the application of calcium. So calcium deficient plants tend to be more stunted, and it's important to know that there's a difference between a deficiency, where it's really hindering production, sufficiency, where it's not going to hinder production, and then more of luxury levels, where you get these quality-driven components. So it's important that your plants might not be showing deficiency symptoms, but it's not kind of thriving. So sometimes additional calcium is really good to drive a lot of more quality components rather than just the lack of disease. But anyways, in a calcium deficient plant, it's going to be, the structure of the plant's not going to be great. So in tomato plant, the leaves, or broadleaf plants more specifically, the leaves are going to be curled over. In your grasses, there's going to be almost like a zippering effect on the leaf. So you can see an example here of corn. Other symptoms of calcium deficient plant. So firstly, in the cereals, you're going to get an increase in lodging because the plant's not going to be structured upright because of those weak walls. So you might get more lodging in your cereals. The other thing, it might be bent over because it's just not that strong, like the actual frame of the plant's not that strong. You'll have, say in tomatoes, great example, you're going to have blossom end rots or a lot of quality problems in your product or in your produce. We're going to have more pest and disease problems as those pathogens can actually enter and access into the plant. And finally, we're going to have less root hairs and less just roots in general. So with the application of calcium, we should have better quality fruit, and that's massive in horticulture because you get paid on a pretty big premium on quality. You're going to have really nice plants standing upright, it's going to look nice and healthy, and your roots are going to look a lot better, a lot more branching, and root hairs and extension. Now, when it comes to calcium actually in the plant, calcium, and we'll talk about this in a moment, it starts in the soil as a ion, so a divalent ion, so there's +2 charge. What happens is this calcium goes up through the xylem to the growing points. So move into the growing points, but because it's lodged into the cell walls, it doesn't move. So once it's stuck in here, it won't move, which means managing calcium is super important to supply calcium throughout the whole system. If this system stops, the plant doesn't have any mechanisms to then relocate that calcium into sink parts of the plant. So then what happens is, say the calcium supply dries up, that's no good. We develop a new leaf, and that leaf is going to be stunted because there's just no more supply. There's calcium in this leaf, which is awesome, but there's actually no calcium left in the sap to then go into this new spot. So this makes it super important that calcium is managed almost from a soil perspective. Foliar applications are great, but it's simply a band-aid to soil problems. We can overcome this problem, but with a foliar application. So we get the foliar on, yeah, this is great. Now we've got heaps of calcium in here, and the plant is going to grow as we want it to be. As soon as we get some more growth come up, that's going to be stunted because we haven't fixed the soil problem. Now, this will be fine, potentially a few applications depending on your crop throughout the season, but really we want to be focusing on our soil health. So again, foliars are great and they do fix the problem, but they fix that problem at that point in time. As soon as you put more growth on the plant, it needs more calcium because the calcium that it currently has is locked up. And we'll talk about that in the second half of the video, how to make sure we're getting continuous flow of calcium. And so I might as well talk about differential sap tests here while we're talking about the plant. What we do in our consulting, it's a major part of our consulting, is we use sap tests and we compare the young leaves with the old leaves. So for example, in this case, we will take this leaf and we'll also take this leaf, get 2 separate tests done, and then compare the concentration. This way, we can actually see the difference, and the relative difference in that mineral between the young and the old, and we compare it to the mobility of that mineral. So calcium doesn't move, right? So if we get, say for example, 1,000 parts per million of calcium in our old leaf, and we're getting say 70 parts per million in our young leaf, this would suggest to me that the plant is not able to pick up enough calcium to go into that younger leaf. So there's a difference between these. That would suggest to me that the supply of calcium is diminishing in the soil, and the plant doesn't have access to more calcium. So this might be worth applying a calcium application. It really does depend on your gross margins of that crop. and how much room you have to be able to apply calcium, because it can get pretty expensive pretty quickly, but it does drive a lot of that quality of growth. What's super important is applying the calcium at the right stages. So sometimes if you've got this example and it doesn't really matter if the plant has a lot of calcium in the plant right now, but it's coming up to a critical stage of influence such as cell division, then it might be really worthwhile applying some calcium. But anyways, the big thing with differential SAT tests is that we want to compare these two numbers rather than looking at a threshold number that says, yeah, if it's past 800, it's fine, because that might not actually be the case. On the other hand, for example, if this is at 500, and this is the first time we're working with a crop, so we don't quite know exactly the mineral concentrations specifically required, we can look at this and go, okay, well, previously may not have had enough calcium, but it's getting more calcium in the plant, so potentially it's on the rise. This just informs us how the plant's picking up calcium. It's important to compare this to your soil test to know, is there enough calcium actually in the soil in the first place? Is the plant picking it up? Because it could be other problems, and we'll talk about those problems later. So it's a great tool that we have in our toolbox, but it's a part of this holistic approach to crop nutrition. If we're at the fruit development stage or fruit fill, we want to make sure we have that continual supply of calcium, because it needs to then get into our fruit. Super important. Foliar applications can fix this, but they are limited, as I said. So let's get into calcium in the soil. Now, this might seem confusing, and it is, so just bear with me, otherwise you can skip. So, calcium. There's three components of calcium really in the soil. There's mineral calcium, there's exchangeable calcium, and there's soluble calcium. So there's three levels. So if you want to see those three different levels, because each mean different things, what you can do is you can get a total SAP test. We do this as kind of baseline for all our clients. So you can get a total test that tells you the amount of minerals in, the total amount of calcium minerals in your soil, as well as the exchangeable calcium and soluble calcium. So for example, in our test, this one, for example, has 770 parts per million of mineral calcium. That will then flow into the exchangeable calcium at 489, and that will flow in at 298. But anyways, if you want to see your soil test in a beautiful formatted report, you can head over to our website. We've got a free soil test analyzer. Chuck it all in, and it's free, so it's cool. Anyways, so it all starts off as mineral calcium. Mineral calcium typically comes from calcium feldspars. This is just a particular mineral in our soil. If you really want to get into it, calcium content increases as you get into the mafic igneous rocks. So, for example, basalt soils have more calcium feldspars in it than, say, sodium feldspars or potassium feldspars, and so you'd expect them to have a higher amount of calcium. Likewise, they tend to be a high pH. Anyways, so there's a large amount of calcium in your soil, in your parent material. Just depends on the parent material, and you can look that up using maps. This is not super available to the plant. We'll talk about getting that available, but in general, it's not available to the plants. It's more available to the microbes. As this weathers, this will release, as well as, say, biological activity, microbes releasing organic acids, that will release into the soil as these clays from the feldspars kind of weather down. Calcium will be as the ion, so freely not bonded to anything, with a loose attractiveness to the negative sites on the clay colloid. What happens here is that this calcium can move in and out of the exchange sites with the water in the soil, where it's available to the plant. So both of these are available to the plant. The plant root comes along, it can either take up straight calcium, the soluble calcium, or it can exchange a hydrogen with this calcium and take it up. So this is pretty straightforward. It's not as complicated as phosphorus or potassium, and simply weathers onto the clay colloid and then can either be taken up from the colloid or into the water and then it's taken up. So this is at a equilibrium between these two stages. To add a little bit more complexity in, you have all these other cations, which are positively charged ions or atoms, elements. So you have sodium, magnesium, you've got potassium, you also got hydrogen and aluminium. So I haven't put aluminium up, but all of these kind of compete for the plant to take up. So if you have way too much magnesium, that's going to antagonize the uptake of calcium. Likewise, if you have too much ammonium, which is basically NH4 positive, that's going to antagonize calcium. Ammonium actually antagonizes the uptake of calcium the most, so it's the most competitive. So this might be quite important if you're applying a lot of ammonium fertilizers, so MAP for example, and then other minerals such as manganese and iron can also antagonize the uptake of calcium. So really anything that has a positive charge in terms of mineral can antagonize the uptake of calcium. So if we're looking at our soil test, we want to know where we need to be on each of these. Again, we like a Albrecht style soil test, so CEC, with the total component. So in our minerals, in the mineral component, we like to see greater than 1,000 parts per million at the very least. Now, typically on a sandy soil, we want to see more than that. This is at the very minimum. So this will go a lot higher just depending on your parent material. Having more than that kind of indicates to us you have some to draw upon. If you don't have any mineral calcium, then it's very hard to draw upon that. So we want 1,000 parts, or at the very least we want 1,000 parts per million of the mineral calcium on our exchangeable. Now this will depend on the amount of clay and organic matter you have in your soil. Typically, the amount of calcium we want as exchangeable calcium will increase with clay content. And that's a really good point. So as I mentioned, the calcium sticks onto the negative sites on clay, meaning that if you don't have any clay in your soil because you've got sandy soil, you're going to have a very difficult time holding onto calcium, which means calcium is going to leach into your subsoil and it's going to be lost. So sandy soils tend to increase the likelihood you're going to get a calcium deficiency. The way to fix that is increasing your soil organic matter content, because soil organic matter has a very similar function in increasing your cation exchange capacity. So increases the amount of negatively charged sites, which allow you to then hold on to your cations. Now the range for the total mineral component, there'll be about 1,000 to 10,000. So anywhere kind of in there is all right. Obviously in a heavier clay, you want more. If you only have 1,000 parts per million in a clay soil mineral calcium, then you've got a bit of a problem. The amount of exchangeable calcium I'd like to see in a sandy soil is about 1,000 parts per million again. So that's quite important. That'll be all the way up to just past 3,000 parts per million in a heavier clay soil. So want more calcium in a heavy clay. It's quite important, just because there's more sites in our soil to actually hold those calcium, or that calcium. Otherwise, it's going to compete against some of the other cations. Now, in the soil solution, we'll be looking at maybe around 750 parts per million, around about that number. Again, it's going to be less for a sandy soil and more for a clay soil. The big important things to consider is your exchangeable calcium, because that's where the plant will access that, and also considering how much calcium you have in your mineral components. Finally, there's another number that we want to look at, and that is the amount of calcium as a percentage on the amount of clay colloids. So for example, if we look at here, and we'll come back to this, there's 10 sites that the cations can stick to. That means we have a effective cation exchange capacity, so ECC, of 10. So there's 10 sites around here. We ideally want our calcium to take up somewhere between 60 to 80%, depending on the amount of clay and everything in your soil, of those sites. Now, this plays into a big concept called base saturation concept, or balancing your soil. There's a lot of arguments, or you'd think there'd be a lot of arguments, and it's very polar in terms of whether or not this is actually a valid way to determine calcium sufficiencies or not. I'm very much on the fence about it. I think to some extent it is important, but it's also not everything. So you kind of want, it's a difficult one, because you don't want to be 100%. All that matters is your ratios. On the other hand, all that matters is whether or not it's efficient, because there's interactions going on, and there's one study that they looked at the amount of calcium and magnesium taken up by the plant is proportional to the concentration, or the ratio of them. So it's a difficult one. So it's important to get it roughly right. I don't think it's important to get it right on. So it doesn't necessarily matter if you've got a sandy soil and ideally it's 60% and you're at 59, doesn't matter. What does matter is that it's roughly right. So if you're sitting at 40% or 50% calcium and you need to be at 60%, and it's slightly acidic, then maybe some calcium applications of lime might be helpful. But anyways, It's important to factor all of this in with all the information you have to make the best choice for your crop. But if you want to see the actual numbers, you can see them here of your texture and the amount of calcium ideally you want as a percentage of your effective cation exchange capacity. Now, a really important thing from that is what's called flocculation. So this is the ability for the clay particles in our soils to stick together. Ideally, we don't want all our clays to be individual, because otherwise they're so small that they cause sealing. So they don't aggregate together, they will seal, and as soon as you get rain, it'll close in all your pore spaces and it will then set like concrete. So we want some really nice aggregation and flocculation in our soil to make sure it can breathe and our roots can explore, and we've got a nice amount of oxygen. So a really important component of this is the amount of cations in your soil. So because your clay particles have negatively charged sites on them, they're attracted to positively charged ions or elements, minerals in your soil. So that is all of these. Now, each of these have a different ability to pull in those clay particles together and cause flocculation. So the flocculation is when you got multiple different clay particles and they come together into a lump or an aggregate. So a really good way to look at this is with the ratios and the percentage of each of those minerals or those cations on your clay colloids. At the lowest end of the spectrum, you have potassium and sodium. If sodium exceeds 6% of your overall saturation, that's called a sodic soil, and it prevents these clay particles from sticking together, which means you get dispersion. So as soon as you get a rain event, that clay is gone with that water. It disperses. The big component of this is the charge of your cation as well as its hydration radius, which is the big difference between magnesium and calcium. So ideally, what we want is we have our clay particles, they're all spaced out. We want to make sure they have something to bind to. Calcium's a great one because it can bring them really in close and it has a large charge. Magnesium, on the other hand, is okay. It's better than sodium and it's better than potassium, but what happens is that the charge of magnesium is actually so great, it's greater than calcium, that it also draws water in. And what happens is when it also draws that water in, it makes the clay then slightly past or pushes that clay further out. Now the interesting thing is that magnesium is actually a smaller element, and it has a smaller radius, which means that the attractive force of it is greater. What's interesting though is that doesn't actually mean it has a better flocculation ability. What happens is that because the charge is so great, it attracts water. And so the hydration radius of this is actually bigger than calcium because it attracts that water. What happens is that this creates a bit of a buffer around the magnesium cation, and then clay sits outside of that. And so magnesium actually has a lesser ability to flocculate your soil than calcium. So calcium, on the other hand, it doesn't have as large of a hydration radius, which means your clay particles can then be drawn in even closer. This is what a lot of people talk about when they talk about the Cal-Mag ratio. Back to that balancing soil concept, it's suggested that a particular ratio of calcium to magnesium will increase the flocculation. And so this is both true and not important at the same time, to some extent. So this is definitely true. Increase your calcium will increase the flocculation of your soil, and there's a bunch of research about that. Where it gets a little bit iffy is that there's also research to suggest that the Cal-Mag ratio does not matter in terms of yield. So I'll explain a little bit more about that just in a bit, but according to the soil balancing concept, ideally in a sandy soil you want slightly more amount of magnesium because your sandy soil is already pretty aerated because there's really large sandy particles in your soil, and so you don't really need that much more aggregation. So you want a 3-to-1, so 3 parts of calcium to 1 part magnesium. In a clayey soil, you want more calcium than magnesium because you just want more of that aggregation together. So again, you can get that on the soil tests. Our soil test report reports on that, so it'll tell you your Cal-Mag ratio. Okay, so you know that for any particular type of soil, there's an ideal ratio of calcium to magnesium. Where it gets difficult is determining whether or not that's actually important. So as we discussed, it's important for flocculation. That's everyone would agree on that. The challenge is now is, does this actually drive production and yield? And the answer is, according to, I think the best research at the moment is it doesn't drive yield. But I actually think there's a lot more questions that we need to ask. So in the study I'm referencing, there's a whole bunch of different farms that they studied. They looked at the yield and they looked at the cow-mag ratio. What they found is that the highest 5 lots of yield ranged from a cow-mag ratio between, say, 2 and 20. That doesn't really matter. So they did that over and over again. Doesn't quite matter in terms of the cow-mag ratio doesn't drive yield. But again, it does help with soil health. So it's a really difficult question. And the funny thing is, with the amount of, say, researchers and science saying it doesn't really matter for yield, but a lot of farmer experience saying it's the best thing ever, you'd think, and such polar viewpoints on this, you'd think that there'd be a lot more research trying to understand what's the go. But there isn't. So the most I've found is that there was some researchers in America trying to test this out, and I don't know if the research is done yet, but it just seems to be, that's really about it. So if you have any research about the cow-mag ratio and how it leads to crop production, I'd love to see it. I'm very much on the fence about it. It's important. I think it's important, especially for flocculation, but it's not the be-all, end-all. And that's kind of where we sit, at least at the moment. And we probably need a bit more SAP data to confirm all of this. The other component is that Ulbrich kind of really popularized this idea. And from what I understand is that during their research, soils were a lot more biologically active, which and suggested that because of that, calcium applications were more important, or you get a better return from your calcium applications. Nowadays, when they redid the studies, there's less biological activity and you get lesser return for your calcium application. So biology and calcium go hand in hand, super important. Potentially with a higher soil organic matter, calcium is super important. And this process also happens, so not with clay, but with soil organic matter. So calcium can bind to your soil organic matter and increase its persistence. So who knows? The other thing with flocculation is that soil organic matter will also flocculate your soil. So soil organic matter has positive sites on it too, so you can increase this. I think it's a component of both. So in short, this is important, but it's not the be all end all. There's evidence to suggest it doesn't quite drive yield, but I don't know if it's looking at all the factors. I don't know if it's looking at quality and health of the plant. So it's one of those things to keep in mind and go from there. It does definitely affect flocculation and soil health, which is important for gas exchange, water infiltration, which you think drives yield, but potentially might not be the limiting factor, or at least in those studies it wasn't. So we'll talk about how to actually calculate your calcium applications using this model in the next part, but you can't quite talk about calcium without talking about pH. So pH, and we'll have a whole another video about pH another time, but we'll go over how calcium kind of plays into pH. pH is simply the amount of hydrogen ions in your soil or your soil solution, and it's made up of two parts. It's made up of the actual hydrogen in your soil solution, and it's also the amount of hydrogen on your clay colloids, because this hydrogen here will recharge this hydrogen if it's lost. So this is the main driver of calcium applications, not so much for calcium itself, but to neutralize that acidity. So calcium is typically supplied as lime. Now we'll talk about other examples of liming of products that can lime your soil and calcium applications, but just in this sense, we're going to be talking about lime. Lime is calcium carbonate. So we've got a calcium there, and we also got a carbonate, which is carbon, oxygen, or 3 parts oxygen, and that has a charge of -2. What happens, you add this to your soil, very small amounts will dissolve over time. So this separates and this separates. Oh, that's a negative. The carbonate will react with the hydrogen. It's a acid carbonate reaction. These two things react, and we actually need 2 of these to react with your carbonate, and that's going to create water, so H2O and carbon dioxide. So that takes out effectively the acidity from your soil. The calcium is beneficial because this calcium will then displace this hydrogen. So the calcium has a greater ability to be attracted to the clay colloid or the sites, which means this calcium will displace this hydrogen, and this hydrogen will get kicked off into the soil solution. Ideally, you want to be applying enough lime for the calcium to displace your hydrogen. The hydrogen will then be displaced for the carbonate to react with it. So over time, this will reduce your overall acidity and increase the nutrient availability of everything else. So some soils have a greater ability to buffer a liming application. The reason why is that more of these sites are filled up with hydrogen, which means you got more hydrogen to displace, which means you need more calcium, or more specifically, need more lime. So you can work out, and we will be doing this in the next whiteboard thing, and how to calculate this. But effectively, if you have more clay and you got a particular pH, you need more lime than, say, you got a particular pH or the same pH in a sandy soil, because you got less buffering. It's super important though that you have this carbonate component to your applications, because so if you don't have that carbonate component, it doesn't react with your hydrogen and doesn't get rid of it, doesn't convert it into water. So if we have gypsum, which is calcium as well as sulfate, the calcium will displace that hydrogen, so it'll come out into the soil solution, but it doesn't react with the sulfate. So you haven't really changed the pH here in much. And eventually, if there's too much hydrogen, it's just going to then displace that calcium component, the calcium cation. When it comes to other minerals, calcium can also displace those other minerals. So for example, if we're trying to achieve a certain Cal-Mag ratio, what we can do is apply that calcium to then knock off, say, either a magnesium or sodium or potassium or whatever other cation. So for example, calcium, if we apply gypsum, which you can do, especially in your sodic soils, the calcium comes in, it pushes that sodium off, and then it takes up that site on your clay. What happens here, especially with gypsum, which is sulfate, then reacts with the sodium and creates a salt, which can then be leached into your subsoil and then ideally away. We'll talk about different products you can use to try and get this right. So before we move on, there's one more thing we need to mention, and that is calcium supply in different depths of your soil. So calcium leaches. It does hold on pretty tight, but over time it leaches. So say, for example, this is our soil here. We've got our topsoil and we have our subsoil. So that's our subsoil. Typically in Australia, a lot of our soils are what's called duplex soils or texture contrast, which means up here is sandy and then down here is a bit more clayey. Typically what happens is your calcium will then leach down over hundreds of years. What can happen is that we have a buildup of calcium in our subsoil. There's an opportunity for this calcium to be taken up by the plant, up into the plant biomass. Say if you've got a cover crop, when we terminate this cover crop, that calcium then is returned to the topsoil and then decomposed into your topsoil. So tapping into this system can actually be a really good way of supplying your soil or your topsoil with additional calcium. The best way to do that is with plants with strong root systems, especially taproots. So you may have heard that some weeds indicate a calcium deficiency. Typically, they have a strong taproot that goes into your subsoil. We'll be drawing up calcium from your subsoil into your topsoil. This here can increase the amount of calcium on your colloids, increasing your Cal-Mag ratio. I'm not too sure if it actually can help you with pH, because even if your calcium displaces the hydrogen, that hydrogen needs to go somewhere. I think very small amounts can leach. It's suggested that microbes can help with buffering pH, but I'm actually not too sure to what extent that can happen. And finally, I mentioned that it can actually go into the plant biomass. The majority of calcium is in the plant biomass. That's not taken off field. So typically, there's not too much of a replacement value you need to supply for calcium because it's not really taken off in your product. A small amount is, but the majority of it is still in your biomass, which means there will be this cycling. The big part of supplying additional calcium, especially lime, is that the plant typically puts out acids and hydrogen ions to displace calcium or other cations off the clay colloid, which means those cations then need to be replaced, and typically that hydrogen then needs to be dealt with say, a carbonate. Cool. So we know that calcium is important for our plants. What options do we have to actually supply calcium to them? So we can almost break this up into more or less soil amendments and foliars. With the soil amendments, we've got a good range of choice. The important thing is that all of these are going to supply calcium. It's what else is it going to supply to? And that can really help guide our decision as well as pH. So lime. Lime typically used to lower your pH. Excellent, 40% of calcium, so a fair amount. It's also pretty cheap. Biggest thing with this, transport costs. When you apply lime, typically anything below a 1 ton application per hectare is not economically viable. So if you're going to make an application at the very minimum 1 ton, don't want to exceed 4 ton, as that will antagonize all the other minerals, because you just apply all this calcium to your topsoil. It's going to antagonize all these things. So no less than 1 and no more than 4. And we'll talk about working out a calcium application. Next is dolomite. Dolomite has a similar liming effect to lime. You also get a bit of magnesium. So for example, if you have an acidic soil and you don't want to increase your calcium content, say for example you're on a sandy soil and you already have that right balance, you might want to apply dolomite instead, because you also get that magnesium and maintain that balance. Next is sodium. Sodium is actually really good for getting calcium into your subsoil because it's very soluble. So this is very important if you have sodic soils or sodic subsoils. This can be very beneficial, as well as getting calcium into soils that have a basic or alkaline pH. Because your lime and your dolomite, they need acidity in order to break down. So gypsum, that's soluble. Will go on, it'll start displacing sodium or magnesium or whatever you need it to do in your alkaline and acidic soils. So gypsum typically is also known as like clay breaker. So this can be really beneficial for increasing that flocculation in heavy clays. Now gypsum is a bit more harsh in terms of its ability to strip off cations because it's all soluble. So I'm pretty sure the upper range for application for that is 800 kilos a hectare, so you don't want to exceed that. So they're kind of your big main three for soil amendments. Next you got guano. You wouldn't typically apply guano as a soil amendment, more as a fertilizer. Excellent amount of calcium in that, 30%, as well as phosphorus and a bunch of whole other minerals. So this can actually be a really good starter fertilizer, especially just getting that calcium into, effectively supplying calcium as a starter. This would be more available than potentially your lime and dolomite. Really excellent source, especially as I said, a starter. Next is soft rock phosphate. Now this is typically used to increase your phosphorus. There's also a fair amount of calcium in there too. So if you're deficient in phosphorus, this is a really good option to also supply calcium. That also has a liming effect as well. So if you need phosphorus as well, this is a great option. There's also a bit of silica in there too, which is very beneficial. All of these can kind of be used in combination with each other in a soil amendment mix. So for example, you can work out how much phosphorus you need as well as a bit of sulfur and then bulk it up with lime to achieve a certain application rate. So you can really mix these together and apply it. It's a great way to do it. In combination with all this, to increase or to make your application go further, you can mix in compost, specifically compost that have a high fungal dominance to it. That will assist with the uptake and storage of the calcium and prevent it from leaching, especially, and that's very important on sandy soils. Next, we kind of have more chemical fertilizers, which are highly soluble and make great foliars. First one is calcium nitrate. Now, if you think back to the nitrogen video, nitrate, there's some problems with nitrate, and it can make low quality growth. So on one hand, you're stimulating high quality growth. The other hand, you're stimulating low quality growth or dilution effect. This can be quite dangerous. So it's important to consider how you're using it. But you get that nitrogen as well. So if you want calcium as well as nitrate, it's also beneficial to apply additional minerals to convert that nitrate into ammonium and then into amino acids, as well as potentially a carbon source with that to help with all of it. Next is calcium chloride. This can be dangerous because the chloride can cause burn if you apply it as a foliar spray. So just be careful of that. I wouldn't use that. The best way to supply calcium as a foliar spray is with calcium acetate. Now this is an organic compound in the sense that it contains carbon. So that can be quite beneficial because it gives that carbon to the plant as that source already. And then if you add additional fulvic acid or something like that, that gives it that extra bit of carbon. So all of these can be applied as a foliar. You can actually get micronized versions of these, so micronized lime, micronized gypsum, micronized guano to be used also as a foliar. Now you need really good agitation in your spray unit to make sure that these don't actually settle in your tank. And the plant actually can take in these as a foliar application. The plant can release acids onto the leaf surface to dissolve these and then take it up. But again, I'd probably go with the calcium acetate if you're going to make a foliar application. So when we talk about applications, we can think about it in two ways. The first one is just applying sufficient levels, and we've talked about that extensively in the other videos. That's simply just taking the ideal level and then comparing it to what you currently have and making up the difference with the application. What we're going to be talking about is more of balancing. So this is working out what we need to supply to make sure our calcium, and it's typical calcium, especially in Australia, typically we have too much magnesium, according to the concept, and we need to apply calcium. So with the balancing, typically we're applying calcium. So we're going to work out the required calcium we need to supply for it to be balanced, and then we'll go into what are we going to apply to make that happen. So first we need three things. We need our effective cation exchange capacity. That's super important. I see a lot of soil tests that have cation exchange capacity, which is not the same as effective cation exchange capacity or total cation exchange capacity. The difference is effective and total, they're the same thing. They include aluminium and hydrogen. So it actually tells you everything. So it tells you all of the colloids versus a cation exchange capacity, which typically will only tell you the big four. So that's calcium, magnesium, potassium, and sodium. So it's incomplete if it's not telling you hydrogen. Hydrogen's massive. We need to make sure, especially if you're going to use this for liming applications, how much hydrogen is in your soil or taking up on your CEC. So we need to know the effective cation exchange capacity. We need to know our ideal level, and you can get that if you use our report, the ideal level that you want it to be at. And then finally, the current calcium percentage. Then what we're going to do is we're going to times that number by 200 because that's the conversion rate between centimoles per kilo to parts per million. And then we'll get our parts per million. So as an example, let's just say we have a soil here, a medium soil with a effective cation exchange capacity of 14. The ideal level at this soil is about 75%. So that's what we're going to be aiming for. And say, for example, we have 50% calcium on our colloid. So the difference between this is 25. So that's going to be 14 times 25% or times 0.25 times 200. And that works out to be about 700 parts per million, ppm. So now what we've got to do is convert this to kilos per hectare. That will depend on what depth you want to go to and the bulk density. I'm just going to use a depth of say 10 centimeters and the bulk density of 1.4. That's pretty easy. If you're just doing it to 10 centimeters, you just times it by the bulk density. So we're going to times that by 1.4. And that works out to be 980 kilos of calcium per hectare. Cool, so we know how much calcium we need to supply for it to be balanced. Now what we can do is go, okay, we need to supply this much. What are we going to use to supply that? If, for example, your pH is sitting at 5, ideally you want to up around that 6.5. What we could do is we could go, okay, well, we have an acidic soil, let's apply some lime to then buffer that out. So we might apply some lime. What we've got to do to now get our application rate of lime is to divide this number by the amount of calcium in the product. So if we're going to do a 100% lime application to achieve this, what we need to do is take that number and divide it by 0.4 and we get 2,450 kilos a hectare of lime. And that would be our liming rate. So super easy to do. It gets a bit more nuanced if you can consider, your X amount of hydrogen, X amount of magnesium, and you can really kind of shift things around. what we can do, rather than doing say 100% lime, we can do 20% of that as gypsum to then get a bit of sulfur in there, or if we're going to add some phosphorus, we can get some of that soft rock phosphate in there. Really just depends on the goals and the rest of your soil test. But that's typically how you do it. Now, 2.5 tons of lime is not a bad application. Again, that's below that 4 tons a hectare, so you just go for it, I guess. So say, for example, our CEC was double and was 28. We would need to be applying 5 tons of lime per hectare. In that case, what would be better is to split that across 2 applications and go 2.5 tons now, 2.5 tons in a few years' time, because you don't want to then start antagonising everything else in your soil. Now, this is a pretty good way to do it. My understanding is that this might even underestimate your total lime requirements. So it's beneficial to take a CEC test every 3 or so years, and we like to use Haney soil test in the meantime, and then go back to an Albrecht style test every 3 or so years to see how we're heading. But this way, we will consider the hydrogen, or at least that empty space, the hydrogen on the clay colloid. Another way of doing this is that required calcium to add is simply just whatever that hydrogen component is, so you're displacing it. And that's in case for liming. If, for example, you got a heavy clay soil and you don't, and it's not acidic, so we're not really liming. What we're going to do is we're going to try and displace magnesium. So say, for example, magnesium is currently at 20. The ideal level is say 10. The current calcium, the calcium we need to supply will be 10. It's the difference in that. So then you can just work it out and then use gypsum to supply that. This is quite complicated. We'll have a whole another video coming out about balancing and going through all of that. But at the moment, in terms of calcium, a simple way to do it is just ideal level of calcium minus your current, times that to your effective cation exchange capacity, and then convert it through, and then work out what you want to apply. Again, you don't need to supply all of this straight up. You can split it, and it's better to split those applications so you don't overwork or antagonize everything else. It's important to note that a lot of these will actually oxidize your soil. So lime, I don't think gypsum will, I'm not sure, but dolomite will, soft rock phosphate probably will, and that can then reduce the availability of, say, manganese, a lot of your trace minerals, so copper, iron, and so smaller amounts more often is better. And then after this, you might just have to apply a maintenance rate every other year. So when we talk about managing calcium in a regenerative way, what we want to do is make sure we have enough calcium in our soil and all the microbes and everything are working so that the plant gets a continual supply of calcium. So we want to make sure that calcium is continuously going from the soil into the roots up to the top of the plant all the time. Every 24 hours, we need a new supply of calcium. So there's a few ways we can do this. The first one is making sure we got enough boron. So boron is required for the uptake of calcium. Typically, you'll find with a lot of the foliar applications, there's boron included, and so that's quite beneficial. But every single time you want to make sure, when we're looking at our differential sap tests and even our soil tests, we want to be looking at our boron if there's a calcium deficiency. So sometimes it's actually not so much about your calcium, but more your boron. So when we're looking through sap tests, we're looking at boron to inform us whether or not it's actually a calcium deficiency within our soil, or it's actually just a boron deficiency. That can depend. So anyways, boron is super important. So the end goal of all of this is not to just continuously apply foliar applications to fix the problem. The problem is that the plant's not taking up from the soil. We want all of our nutrition to come from the soil eventually. So we want to improve our soil environment using microbes and a bit of nutrition to supply all of that. So once we kind of nailed out boron, making sure we've got boron, we want to really start focusing on our biology. So that's things like the rhizophagy cycle, which will actually supply calcium to the plants. So there's calcium in the cell walls of their microbes as well. That will supply calcium to the plant. We're going to add mycorrhizal fungi. Mycorrhizal fungi also is important for the uptake of calcium. So there's a few studies there you can look at. Super important. Next is calcite solubilizing bacteria. Similar to all our other solubilizing bacteria, there's some bacteria out there, and you'll see a list here from research. They can actually solubilize calcite, which is calcium carbonate in the soil. Super interesting. It allows us, when we're then looking at our soil test, we've got a heap in our total component in our soil test, so total calcium. Can we transfer that into exchangeable? And our solubilizing bacteria will help with that. So really, we want to get these numbers up. The way to do that, as with everything, we want to make sure we're increasing the photosynthetic ability of our plant to then feed these microbes with more sugars. We also potentially soil primers. We've got a video on that. You can check it out. They're limited. Root exudates is where it's at. So increasing the root exudates from the crop itself, but also before that with the cover crop or companion crops or something like that. Next is actually adding that calcium to the soil, which is, I guess, more of this idea. Sometimes you don't need to overdo it. If you're just applying, say, guano as a starter fertilizer, and we're getting that calcium in there, that's a really good source. Actually, it's good to note, Super also has a fair amount of calcium in there. Not that you'd use it so much as a calcium source, but there's calcium in your Super, so it's potentially beneficial to think about it like that. Next is deep taproots. So the idea with this one is that we'll be cycling calcium from your subsoil. It's not quite accessible to a lot of your crops. We're going to take it up with a taproot into the biomass of the plant. That plant biomass is going to die and then it's going to accumulate in the topsoil. That's a good way of then cycling that nutrients in the soil. And for that, we need plants with deep taproots. Cover crops are excellent. You can really get a variety of taproots in there. And then in addition to that, and I haven't written it up here, is increasing the soil organic carbon component in your soil. So this is super important, especially in sandy soils, to hold on to that calcium so it doesn't leach. Super beneficial. Building up that reserve, especially in your sandy soils where you're going to run into a deficiency sooner. Building up that reserve in your topsoil can really help with that. And then finally, while we're doing all this, we're going to fix the in-season problems with foliar applications. Ideally, we're going to be applying our calcium acetate. It's just easier and it's better for the plant. But the idea is this will just kind of push the crop along until you can get everything else working in your soil. We don't want to be stuck on foliar feeding forever. It's really just in the next few seasons while we're trying to regenerate our system. So as with all our minerals and managing them in a regenerative way, it ultimately comes down to making sure that mineral is first in the soil, making sure it's available, making sure we have our microbes of fungi and plants interacting with each other and with the minerals to then supply that. And then if that's not happening, we fix it in the short term with foliars. And that's kind of the big idea with regenerative nutrition management. So with this, once we start getting constant supply of calcium, that's really where the deficiencies come from. It's where that supply cuts out, because again, if the supply only goes to that leaf and then stop, that calcium in here won't be able to then relocate into younger leaves. So we need to make sure it's continuous, happening all through the season. If we don't get that, that's when we start to get diseases. That's when we start to get all of our, a lot of the fungal diseases, as well as our pests. And most importantly for the horticulture guys, it's where we get a lot of post-harvest diseases. And you don't want those because it's going to really start degrading the quality of your product and your premiums. So there's one study they're looking at calcium chloride applications actually, which is like, I guess more or less the worst, not the worst, because your calcium nitrates is going to increase your nitrate concentration, and we don't want that because that's what your microbes, your diseases and pests are looking for. But anyways, calcium chloride applications as dips in post-harvest, and that was able to reduce or increase the storage ability of the plant, of the fruit, as well as in-season calcium chloride applications also reduced storage diseases. So super interesting stuff, especially when we start getting into quality. When we're thinking about the fixing calcium with foliar applications, what we can do is target our applications to critical points of influence so that we get the biggest bang out of our foliar application and our money. So for calcium, that's super important for firstly bud determination. So that's when the buds or the head of say cereals are determined, and that can actually be either, so for our cereals, it can be up to a few weeks, I think it's up to 3 weeks after germination. So it happens super quickly in our tree. Sometimes that can actually happen 2 years beforehand. So in say, a vineyard, bud determination happens in the previous season. So you've got to be thinking a fair way out sometimes with these things. But calcium is required because we're getting that cell division. We also want it during the stage of kind of putting that mass onto the plants, the photosynthetic ability of the plant. Now, this is more important for the quality of the growth so that we have a nice strong plant and it's going to be able to photosynthesize as much as it can to then put that into grain. So the photosynthetic ability of the plant, that's quite important. Next is flowering. So it's important that the calcium's there to support the reproductive components of the plant. Next after that is cell division. So between 10 to 14 days. So as soon as the plant is fertilized, the flowers are fertilized, the embryo will undergo rapid cell division for about 10 to 14 days. That's super important to have lots of calcium supplied during that stage so that we get as many cell divisions as possible. After that, now after this one is our grain fill stage. Now we actually don't want as much calcium in there because that's going to antagonize our potassium. So we want to make sure potassium is now dominant in that stage so that we can transport those carbohydrates into the either fruit or grain. So super important. We don't quite want to be supplying a lot of calcium after that. We still want enough during grain fill because that's going to help with this the cell integrity of the plant. So if it's looking like you're getting a deficiency, definitely apply it because there's no point in having all this big fruit if the skins are going to be soft and just break apart in your post-harvest. So that's all about balance in your grain fill, but really potassium is the main driver for that critical stage. After that we have ripening, which doesn't necessarily need calcium. So really we've got three big ones. We got bud determination, photosynthetic ability somewhat, other minerals more important, but it's also important for just that quality in the frame of the plant, flowering, cell division. Super important. So if we can time our applications to really consider these ones, bud determination, especially for our cereals, that's really going to be coming from our soil. So this side of things, especially potentially our starter, so it's guano, and our microbes. So getting a seed coat onto, including microbes and fungi. Foliar application, foliar application, foliar application. And making sure we're adding boron. We can take differential sap tests to know actually where we're at until the system has improved itself so that we don't need to do it. Awesome. Well, if all of that sounds great to you, it sounds great to me, it might be worthwhile for you to sign up for a free consult with us. What we'll do is we'll sit down, have about 30 or so minutes to discuss all of these opportunities and how we can help you transition to your farm to regenerative without that loss of yield. We're big on the science behind nutrition so that we can actually transition our farms in a low risk manner without losing yield. So we don't want to see farmers transitioning to regen ag and you get this drop in yield. We want to make sure it's maintained and then improving. So if that sounds interesting, you sign up for a free consult. Otherwise, thanks for watching. My name's Teal. Cheers.